Good evening, everyone. Thank you for coming tonight. My name's Trent Garner. I work here at ZSL in the Institute of Zoology. And along with Kieran Bates, who's hiding over there, we organized this symposium for the Holobiont Center, the Leverhulme-funded Holobiont Center. This is actually the 9th and final Science and Conservation event that we've held for 24-25. So thank you for coming and wrapping up a wonderful series of scientific events. I'm going to introduce the director of the Leverhulme Center for the Holobiont, and I would like to say I think it's pretty safe that this subject tonight is a little bit outside what you normally hear from the Zoological Society of London. Normally you're here seeing talks and presentations about rather large things, and this is one that's focusing more on microscopic organisms and their relationships with the larger things. I hope by the end of tonight you'll see there's a reason why the Zoological Society is investing in this subject and why we're partnered in the Leverhulme Center, because, as you know, we're an organization geared around science and conservation, and one of the emerging themes is the relationships between the large organisms and these microscopic organisms should be something we should be embedding into our conservation planning. But without further ado, I'd like to introduce the director and the person who actually spawned the Center for the Holobiont that the Leverhulme Center has funded. It's Professor Tom Bell from Imperial College. He's been deputy head at the Department of Life Sciences at Imperial College London. He joined Imperial College as a lecturer in 2011 and concurrently held a Royal Society University Research Fellow from 2010 to 2018. He was a departmental lecturer at the University of Oxford and Beale Tutorial Fellow at St Hilda's College, University of Oxford. His BSc honours comes from the wonderful University of McGill in Canada, and he also holds an MSc in zoology from an equally wonderful university in Canada, the University of British Columbia, and his doctorate of philosophy in zoology comes from the University of Oxford. Tom, if you'd like to come up.
Thanks, Trent. So you get two Canadians to start off with. Sorry about that. We didn't plan it intentionally, I don't think. Thanks very much to Trent and to Kieran for organizing today. It's a really great opportunity to tell you a little bit about what we've been up to at the Leverhulme Center for the Holobiont. And I thought I'd start out today with my 5 minutes of allocated time by giving you a snapshot of all of life on Earth. So this might not be a familiar diagram to you, but it's one that I challenge my undergraduate students with. It's with the wonders of DNA sequencing, we can actually start to put together a picture of what all of the tree of life looks like on Earth. So we can take a little bit of DNA from every single organism that we discover. We can look at shared pieces of DNA, and we can use that to construct a family tree, if you like, of everything that exists. And this is many, many years of research, lots and lots of people doing this kind of thing. And this is what results. The branching pattern shows you which organisms are closely associated to each other and which ones are more distantly related to each other. And the challenge that I usually set my students is to ask them where they lie on this tree of life. And you've had a minute or so to look at it, to wonder which great branch of the tree of life humans might fall on. And where they actually fall is just down there. And that red branch there is not the human branch. That's not us. That's actually the Episthecons, which includes all animals and all fungi, in fact. Everything that's familiar to you. The land plants are down there as well. So there you have pretty much all of the organisms probably that have been presented in this series over the last, I don't know how many years. So the vast majority of life on Earth, the vast, vast majority is microbial. It's the sorts of organisms that are completely unfamiliar to you, and until very recently were completely unfamiliar to science. And what we think is that if you look at any particular organism, you can see this vast diversity of microbes which are living on top of it and inside it and interacting with it in very important ways. And we think that probably if we want to understand any particular large charismatic organism like this one, then we probably need to start to try to understand how it's interacting with those microbes. And that's where this idea for the Leverhulme Center for the Holobiont came from. It's this idea that we can't understand organisms except in the light of the microbes with whom they're interacting. So a holobiont is just this combination of a multicellular organism and all the sorts of microbes that they're interacting with. Some of those microbes may be pathogenic, they might be harmful, some of them might be beneficial, do good for the organism, and some of them may be benign. And what we're trying to do is trying to disentangle those sorts of interactions. This is a large-scale collaboration between amongst a number of different organizations, including the ZSL and other places like Kew Gardens and Cabby and others, the Natural History Museum. I myself am based at Imperial, and we have 3 different faculties which are involved in the center in that capacity. It's a project that has been funded over 10 years. We're just in the going into the 3rd year of this center at the moment, so we still have quite a long way ahead of us. And I'm very happy to give you a little bit of a flavor of the sorts of things that we've been doing in the center, both from those of us who are working internally within the center and with the special guest, Sarah Knowles, who will be talking a bit later about some of the work that she does. So our first speaker, I'm glad to say, is Phil Jervis. I won't give you a lengthy introduction to his career, but he's able to do that himself. He'll be talking to you a little bit about frogs and the microbes that they interact with. Thanks very much.
So I'm a postdoc at the ZSL Institute of Zoology here and funded through the Leverhulme Holobiont Centre. I'm just going to be giving you a quick talk now on an overview of the work that I've been doing so far on this postdoc, how we've applied this knowledge to amphibian conservation, and looking forward to kind of where we want to kind of move next in terms of the science to support the conservation being done. So my research is largely focused upon a single amphibian disease, chytridiomycosis, which is caused by the Batrachochytrium chytrid fungi. And these have been implicated in the declines and extinctions of over 500 amphibian species worldwide. Many of these declines are driven by a single pathogen, Batrachochytrium dendrobatidis, which I'll call BD from now on to save myself going mental. Following the global spread of BD, and especially the BD GPL lineage out of Asia, it caused the collapse of the susceptible amphibian communities around the world. And even though there's the headlines of these amphibian kind of disease collapses and population collapses, not all species and populations within a species were affected equally by the disease. So to understand what's driving these differences in disease outcomes in populations and between species, we need to understand about the interactions that take place upon amphibian skin. So BD is a fungal pathogen of amphibian skin, and they kind of infect frogs through aquatic zoospores that either spread kind of like through water and swim through, or through contact between an infected host and another animal. Once the zoospore comes into contact with the skin, it then embeds within the skin, and as you can see in this inset picture in the top corner, the dark purple sort of sporangia, which are these kind of like reproductive stages of BD, then create more spores, which are then emitted. And these emitted spores can either go on to infect a new host and transmit the disease, or they can also, the disease itself progresses in a single animal by these cycles of emitting spores and reinfecting the same host. And so the main defense that amphibians have against this pathogen is actually the mucosal layer on the skin and the cocktail of antimicrobial compounds that are present. And these antimicrobial compounds are both produced by the host and by the skin microbiome as well. And so even though there's like a massive diversity of chemicals present, my work largely focuses upon one set of immune defense, like antimicrobial peptides or AMPs. And these AMPs are known to inhibit BD growth, but also to shape the microbiome and hence the antimicrobial compounds the microbiome produces on the amphibian skin and the immunity of the host. So the first piece of work I'm going to share is kind of a natural recovery from a chytridiomycosis outbreak in Europe. And this work focuses upon a single species, the common midwife toad, Aletes obstetricans, which is widely distributed throughout central Western Europe and has experienced severe declines in high elevation areas and even within protected areas. It's a focal study organism within the amphibian disease research groups within both Imperial and ZSL. Despite the sort of severe local declines in these places, some of these populations are still persisting with the disease and they're even showing some signs of recovery. And this makes it an ideal study organism for looking at the immunity and the associations between these recovering populations and these populations that are heavily affected by disease. So steering away a little bit from incredibly dense science, I'm then going to look at the conservation implications for this work and some of the work I've been doing with the Smithsonian in Central America. And the story of BD in Central America is that during the 1980s, there was a wave of the pathogen that passed through up from Costa Rica and through into Panama. And as this BD wave spread through, there was the collapse of many susceptible amphibian communities, and conservation groups at the time were going ahead of where this wave was spreading, collecting animals that they thought were going to be susceptible to infection, and took them into assurance colonies, where they're kind of kept in zoological collections waiting for reintroduction. And although these ex situ populations are doing well, one of the problems still remains that to be able to be reintroduced, the pathogen is still present within the communities they want to reintroduce them to. So the project we're working with in the Smithsonian, the Panamanian Amphibian Rescue and Conservation Project, has 12 species held within these ex situ assurance colonies. And we have active projects across 4 of these species. So 2 of these species, the crown tree frog and the horned marsupial frog, we're in very early stages of this project, and we're currently just generating baseline data on their immunity. So what are the antimicrobial peptides they possess, and what are the bacteria present in their microbiome? For the 2 other species we have here, the lemur frogs and also the harlequin frogs, we have a bit more of a advanced into the project, and they've actually started to do reintroduction trials now to assess the ability of these captive animals to be reintroduced. So looking at the first one, the lemur frog, lemur frogs are one of the primary taxa that we're working with in this collaboration, and I've carried out field work with this species over the past 3 years. And while lemur frogs initially declined across as BD spread through, there have been signs of population level recovery within protected areas. However, due to habitat destruction, these areas are now very isolated from each other, and so reintroduction is still going to be needed to repopulate some of these suitable habitats where the animals haven't managed to persist with BD. Not much is known about the immunity and the disease ecology of these animals in the wild, and outside of characterization of 2 antimicrobial peptides that are shown below. Gonzalo Rosa at ZSL is leading a wider project on lemur frog conservation across Costa Rica, Panama and Colombia. And my part in this project is to characterize the differences in AMPs and microbiome between recovering populations of wild lemur frogs. And it's important to understand the mechanisms by which these populations are persisting with disease, and also a secondary objective to kind of assess the animals in captivity and whether they're fit to be released back into the wild. And finally, there's a lot of people that have helped me with all of this work. As I said, thank you to everyone. And I guess, any questions? Oh, no, not any question. That comes later.
Yeah, what we actually do at the end is we'll have everybody brought up here, and then you can ask some questions collectively as a group. So you can really skewer them with all the ideas that you've come at watching these talks. Our next speaker was supposed to be Dr. Elliot Murphy, but he's unfortunately suffering from dysbiosis of his microbiome. He's got an infectious disease right now, so he's in no fit condition to come out, and he would probably end up transmitting microbes to all of us. So he's said no, and so what we'll do is we'll move straight on to Dr. Theo Llewellyn at Imperial College and the Natural History Museum. And Theo's research uses a combination of genomics, phylogenetics, and ecology to understand the evolution of fungal symbiosis, so moving away from bacteria into the fungi, and in this case, fungi that are not actually killing amphibians. In their current position as research associate at the Leverhulme Center for the Holobiont, they are working with Professor Alfred Vogler to develop bark beetles and their associated fungi as a new system to explore holobiont evolution and diversification. Before this, Theo completed a PhD at Imperial College and the Royal Botanic Gardens at Kew, where they studied how lichen-forming fungi adapt to survive in extreme environments. Thanks, Theo.
Okay, So yeah, I'm going to be speaking about the research that I've been doing over the past couple of years, looking at how we can describe bark beetles within this sort of holobiont framework. So if you go and look at almost any sort of conifer tree, and you kind of look up closely at the bark, it's not super clear here, but you can kind of see some like black holes. If you were to peel back the bark, you can see this kind of maze of tunnels, which are created by bark beetles. So bark beetles will kind of tunnel a little hole into the bark, and then they kind of spread out, and they lay their eggs under here, and their eggs hatch into larvae, and they kind of rear them. If you look even closer at the sort of the walls of these tunnels, you might be able to see some sort of like white fluffy material, which are fungal hyphae. These are kind of microscopic fungi, so not the sort of classic mushroom-forming fungi that you might know already. So if we're thinking about it as a sort of holobiont framework, who are the players in this holobiont? So from the beetle side, you have two groups of weevils, the platypodids and the scalitids. From the fungal side, they're mostly, as I mentioned, microscopic fungi. So you have some kind of single-celled yeast, but more sort of filamentous fungi. And perhaps the most well-known of these bark beetle symbionts are called the ophiostomatoids. And they are what cause these kind of blue staining that you often see in timber. From the plant side, equally diverse, so almost 200 different plant families have been associated with bark beetles, and it kind of depends where you are in the world. So in the tropics, the most common hosts of bark beetles are dipterocarps, but some of the kind of most well-known hosts are conifers, because that's where bark beetles really have their strongest impacts. So who sort of wins in this game? Well, most of the time they play pretty fairly. These bark beetles tend to only target dead or dying trees, and they sort of form a part of the natural cycle of kind of recycling nutrients, opening up spaces in the forest to allow new growth. But under certain conditions, climate change being one of them, you kind of have these mass outbreaks of bark beetles where they're able to destroy huge areas of healthy forests. This also affects us directly, so bark beetles are able to attack a lot of plants that we care about personally. So avocado at the moment is facing really kind of strong pressure from bark beetles and the fungi that they bring with them. And then also for the timber industry. So a lot of these fungi, as I mentioned before, are known as blue staining, so they produce a lot of melanin. And this kind of stains the bark, and it prevents it from being used for certain purposes. So how are we studying this quite complex system? These beetles are tiny, they're just a few millimeters long, they're very diverse. You find them in lots of different habitats, and the fungi are obviously even smaller, very diverse again, and they're quite hard to find. So the approach that we're using is called metabarcoding. I think that's my one joke, so I'm glad that people laugh. So the good news is bark beetles are already actually doing a lot of the sampling for us. So they collect fungi either kind of on purpose or just by sort of brushing past it, and they take it between different trees. They carry these in either kind of specialized organs or just kind of on the surface of their exoskeleton. So what we use are passive samplers set up in forests that trap beetles as they're kind of traveling from one tree to another. We then do DNA metabarcoding on the whole beetle, sequencing all the DNA, and then just pulling apart which DNA belongs to the fungi. So I'm going to present two case studies of how we're using this approach to study bark beetles. The first is focused on the tropics, and the second one is based here in the UK. So for this first one, we looked at around 500 bark beetles collected from two sites, one in Borneo and one in French Guiana. And we wanted to see what are the sort of main factors that drive the fungal diversity associated with these beetles. So another PCOA plot, sorry for that. But the main thing to look at here is the colors of these points. So each point is a bark beetle, and if points are close together, it means that their fungal microbiome is very similar. So if we color code them according to which beetle host group they are a part of, you can see that the kind of blue and orangey color kind of all sort of overlap. They're not really very well separated, suggesting that these two groups of beetles have quite similar fungal microbiomes. But when you color code the plots based on the site that they were collected in, you can see that the two areas separate quite clearly. So it's suggesting that the fungal microbiome of these beetles is more strongly affected by where we collect them rather than which beetle group we're looking at. And then when you look at sort of which fungi are driving these patterns, in French Guiana, there's an abundance of this group called the Dithydiomycetes. So these are often associated with leaves, things like endophytes, sort of beneficial fungi, but also plant pathogens. Whereas in Borneo, there's a dominance of the Agaricomycetes. So these are often associated with wood decay. So just by using this metabarcoding approach, we can already kind of tell something about the ecological patterns of these two different areas. So for the second project, a bit of background. So in 2018, the Forestry Commission detected an invasive bark beetle in the south of the UK, in Kent. So it's this species, the eight-toothed spruce bark beetle, Ips typographus. And this is one of those kind of really kind of damaging pests. So they're able to wipe out really big areas of forest. And this previously wasn't present in the UK. So they did a really big effort to try and kind of contain this. They burned the areas of the forest where they detected it, sort of really tried to isolate it from spreading. But unfortunately, it wasn't very successful. So these populations of Ips typographus are still here. And quite worryingly, they seem to also be targeting native UK spruce as well. So it's not just the kind of plantations where they often are found, but they're kind of spreading into native UK plant species. So what we wanted to know, so we know that Ips is here. We wanted to see whether Ips are also bringing with them new fungi. So to do this, we first had to figure out which fungi were already here in the UK associated with bark beetles. So we built this sort of Symbiont library. So we collected data from lots of different sites all over the UK, and we did the metabarcoding of these beetles. And I'm not going to kind of go into these results really, but it's just to show that we now have a lot of symbiont data from different countries, from different native UK beetles, also from different types of forests as well. So we have quite a good idea of what was already here. We then compared that to the fungal DNA that we get from these invasive Ips beetles. And so when you do this metabarcoding, you tend to get like a long list of names. Some of them aren't identified fully, and it doesn't really tell you much about the biology of these fungi. So this is where trees of life come in very useful, because we can place our Ips fungi onto trees of life and then predict what their biology is going to be based on who their kind of closest neighbors are. And so using this approach, we've been able to first already spot some potential invasive UK pathogens. So for example, we've seen that this Ips is carrying a really damaging fungus called Endocnidiophora polonica, which is not currently being monitored as a pathogen within the UK. We're also able to track known pathogens. So we see that Ips are also carrying the causal agent of Dutch elm disease, Ophiostoma novo-ulmi. That's something that's already in the UK, but we can see that also these invasive species are carrying it. But perhaps what I think is the most interesting part of this method is we can also predict potentially new pathogens. So there are 4 other fungi associated with this beetle that are quite closely related to the cause of Dutch elm disease. So we can look at these to potentially suggest new fungi that might be a threat to UK forests. So to conclude then, hopefully I've demonstrated that metabarcoding can kind of give us a snapshot of the fungal diversity in an area, which we can use to look at biogeographic or ecological patterns. Unfortunately, these UK invasive bark beetles do seem to be bringing in new plant pathogens. But perhaps on a more positive note, by combining metabarcoding with these trees of life, it can take us beyond just naming which fungi we have, and we can start to suggest what these fungi might be doing, whether they're a risk to our UK habitats, and we can also plan and think about potential actions for controlling them. So thank you for listening. This is the team that works on the bark beetle projects, led by Professor Alfred Vogler, and I'll be really happy to take questions after the Q&A. Thank you.
Well, it gives me great pleasure to introduce our keynote speaker for tonight, Dr. Sarah Knowles. She's an associate professor of ecosystem biology at the University of Oxford's Department of Zoology. She completed a BA and DPhil in Oxford before becoming a postdoc at the University of Edinburgh. And her current research at Oxford focuses on the mammalian microbiome in the wild. Using wild mice and other rodents as model systems, her group studies both what shapes the microbiome in natural settings, but also how it impacts the host. They do this by combining field studies, experiments, computational, and comparative approaches. Thank you for coming tonight, Sarah.
Thanks very much, Trent, and thanks for the introduction. It's great to be here. Let me just find my first slide. So with apologies, as Trent said, I mainly study mice, not the most sort of charismatic species or species of conservation concern, except I suppose they are one of the most invasive species, successful invasive species on the planet. So they're absolutely everywhere in the world. But hopefully the reason why we study them will become clear, because they are a really important model organism. They are the mammalian model system. And so by studying them in the wild, we have a lot more tools to be able to understand kind of fundamental aspects of microbiome biology. Okay, I thought I'd start this evening with a question, which is, how human do you feel tonight? Does anyone want to... Feeling good? Feeling as good as this lady? She looks pretty good. I'd say, I'm feeling pretty good, maybe 80% human tonight. But actually, if we kind of think about how many bacteria we have inside us, depending on how you count and what you count, you're not very human. So if we just think in terms of the number of cells, the most recent estimates suggest that we have about 10 to 13 human cells in our body, and we have about the same number of bacterial cells. So if we go on cell number, you're about half human. Now, most of the bacteria that we have in our bodies reside in your colon, so that's where about 98% of them reside. So that's why I guess lots of people are very interested in the gut microbiome, as am I. So by cell number, we're about half bacteria, but if we think about all the different genes and contained in the genomes of those bacteria, it becomes a bit more staggering how how small the percentage human we are is. So how many genes are there in the human genome? There's about 20,000, roughly 20 to 30,000, depending how you count a gene. But if you look at the number of different microbial genes in your body, it's upwards of 2 million. So if we look at the genetic sort of functional potential in our bodies, at best, 1% of that comes from your genome. At worst, maybe 0.01. So they have this tremendous genetic potential, the bacteria in your body. And I think it's even more striking when you think about how little mass they take up. So yes, 50% cells, but when we think about the weight of those bacteria, they're only making up about 0.3 or 4% of your body weight. So they're really, really packing, they're punching above their weight, I guess, in terms of what they can do for their size. So I guess it's unsurprising then that in the last 20 years, scientists have discovered all the different ways that these bacteria in our gut influence our biology. So perhaps most obviously, we know that they help us digest our food. There's lots of things that we struggle to break down, complex plant polysaccharides we struggle to break down without them. They're also really important for the development of our immune system. So there are whole subtypes of immune cell in your body that just won't develop if you didn't have a gut microbiome. Of course, we've heard they're very important in pathogen resistance, and that's the same in the mammalian gut microbiome. They have all sorts of metabolic effects. So, for example, regulating how fat you get. There's a well-known role in obesity. They're making different molecules that we can't make ourselves, and they're even shaping our behavior. So, a recent study in mice, for example, showed that the gut microbiome influences the motivation of mice to exercise. So, they have all these many, many effects. And that's why, of course, there's been a sort of explosion in interest in the gut microbiome, and lots of pop science books and recipe books about what they're doing and how we can modulate our gut microbiome for improved health. But I would say microbiome science is also a fairly young field, really. 20 years is not an extremely long time. That's sort of when we started characterizing these communities as a whole. And there's lots we don't know. And I would say for mammalian gut microbiome, most of what we do know comes either from studies in humans or from lab animals that are housed under conditions like this. So this is a kind of typical animal house where you've got maybe 5 mice living in each of these cages, all males or all females, and it's all very sterile. And these mice have been typically bred, so they're inbred, they have reduced genetic diversity. They've got no environment, really, have they, in terms of what's normal? They've got not much social environment. It's all a bit weird. And they've been kind of what we call re-derived. So they've been made sterile with lots of antibiotics to get rid of pathogens, but that means their microbiome's also been altered. So studying them in this environment has a lot of challenges and is extremely different from the kind of environments that these animals would have in the wild, which are a bit more complex. So something like this. So this is a wild house mouse. Hopefully you can see him down here. At one of my study sites, which I'll talk about today. So, oh, there's a weird noise. So it's I just show that to keep you all awake. But yeah, so no mice were hurt in the making of this video. There is a ledge down there. You can't see it, but anyway. But obviously, there's a lot more challenges here. There's a lot more microbes around. You've got to deal with obviously quite a lot of weather. It's challenging. You might get predated, although in this site there's not actually that many predators. So what my lab is trying to do really is try and understand 2 questions. First of all, why do gut microbiomes vary in natural populations? What are the main forces that are shaping their composition? Because that might be quite different from in a lab. And I guess the thing I'll talk more about today is, what are these gut microbiomes actually doing to animal physiology? They're extremely variable in nature. If you go into one population, there'll be a massive amount of variation among individuals, but does this really shape physiology and ultimately host fitness in a natural environment? It looks like they're really important physiology in the lab, but that, as I said, that's kind of a strange environment. So the way that we've been doing this, so I picked mice for a reason, it's because they are this model organism, and what that allows us to do is to study them in a kind of completely natural environment and make field observations about their microbiome, about their environment. But one big challenge of kind of studies in these wild populations is that you can't do experiments, and that's really important for getting at causal inference. So we can have correlations between the microbiome and some phenotype. But it's very difficult to prove that the microbiome is causing that. So, of course, the nice thing about mice is that we can also do lab experiments where we can manipulate the microbiome to see what it's doing. And one of the classic experiments that people do in microbiome science is to do a microbiome transplant. You may have heard of fecal microbiome transplants. So this is the kind of experiment you can do where you take a microbiome, plonk it into ideally a germ-free, so a sterile animal, and look at whether the trait that you're investigating is shaped by that microbiome. So what we have been doing for the past, only a couple of years, is bringing microbiomes from the wild, transplanting them into these sterile organisms to see what does this natural variation actually do. Okay, so to do this fieldwork, obviously we need to go to an amazing place, because otherwise why do biology? So we do our fieldwork on this island, which is off the coast of Pembrokeshire, called Skocum. I don't know whether any of you have ever known about this, but it's sort of near Scoma. If you go to the far west of Wales and keep going, a few miles offshore, you'll get to Skocum. It's about a mile long. It's basically a rock in the sea, but it does have house mice since about 1890-something. It just makes a really nice natural laboratory. So if we study house mice on the mainland, there's often pest control, there's predators, there's competition, a complex array of selection pressures. So we've chosen to study them on an island, a bit like people have studied, finches in the Galapagos. Pick an island, it's a nice sort of simple system. So we go out to this island 3 times a year we've been going for the last few years and catching mice and studying their microbiome. So this is kind of just a picture of one of our traps that we set. So we set these live traps, they're called Sherman traps, all across the island. I'll show a map in the next slide. This is trapping point 182. And the mice live all over the island. So here's a kind of aerial view of the island. We've got two major regions that we trap in. We trap down here. There's this they love it down here. This is kind of like Mouse Metropolis, this rocky area called the Quarry. And then there's actually a bird observatory on Skocum. That's the reason why we can visit so easily. So they also live around the buildings. So there's a bit of variation in their lifestyle. Some are more commensal, the ones that live in the kitchen bin over here in these buildings. Some pictures somewhere, yeah, so this is the observatory buildings. Some live on dry stone walls, just kind of in the cracks and things. Others live kind of on the cliffs, often in disused seabird burrows. And this is the quarry down here where they kind of live, in burrows behind these rocks. So when we go trapping, we set about 150 traps every night. And it's very posh fieldwork conditions. We sit in the garage. This is Evelina, the first PhD student who was brave enough to follow me out to this island. So she's sitting there. Once we've caught the mice, we then take lots of measurements to assess their age, their sex, reproductive state, and so on. And we mark every single animal with a pit tag, like a pet chip, so that we can individually identify them. And also monitor them through a system of loggers that we have on the island to measure who's where, when. And then as soon as possible that day, we let them go again exactly where we found them. And of course, we need to measure the microbiome, so we collect poo. So this is what I've built my whole career on, is collecting mouse poo. But it really is gold dust. There's so much you can learn from mouse poo. So we take this from the base of the trap, and then we sterilize all the traps so that we don't get cross-contamination. So this is me back in 2019 with the first mouse we ever caught. And now we've caught about 745 individuals and got about, over 1,600 samples from them over the last 4-ish years. So one of the first things we wanted to look at in this system then is just to see, what does the microbiome of these wild mice look like compared to your average lab mouse? Because I think it's important to understand, whether lab mice are a good model for wild mice and what the differences might be. So what Evelina did in the first bit of her PhD was just to compare the two. So she had some lab mice from our lab house in Oxford, and then she had our wild mice, and she got lab mice from several different colonies and wild mice from Skocum and other islands that have also been invaded by house mice. And if you look at a kind of broad scale, so just the bacterial phylum, they don't look that different. So broadly, the same 4 phyla occur in lab and wild mice. But if we break it down to kind of species level, and here's another one of those lovely principal coordinates plots, you can clearly see that the lab mouse microbiome and the wild mouse microbiome are easily distinguishable. So they do have some overlapping species, but they cluster very distinctly. And these light blue ones are the ones from Skocum. But she also looked in a bit more depth at what the differences might be. So we typically find that the wild mice have a higher diversity of microbes than the lab mice. Probably some of them microbes have been lost when they've been domesticated in the lab environment. They're more variable among individuals, so within a population you get lots of different types of microbiomes, and they're more dynamic in the wild. So I guess all things you would expect. This plot just shows you how they're more dynamic. So if we take 2 samples from any given mouse and look at the relationship between how far apart those samples were taken, so the days between the samples, and this is a measure of how different the microbiomes are. It's called the Jacquard distance. It's essentially the proportion of bacterial species that are unique to one of those samples and not shared. You see that in lab mice, the longer the time between 2 samples, the more different the microbiome, but it sort of drifts upwards quite slowly. So there's kind of this slow turnover in microbes within an individual. In the wild, we see this very rapid plateau, and you kind of, after 20 days, your microbiome samples are about as different as they're ever going to get, and they kind of stay there. So much more dynamic microbiomes in the wild. What's also been interesting, though, is that not everything has differed between lab and wild mice. So we also looked at the patterns of microbiome assembly in lab and wild mice. So obviously in the lab it's easy. In the wild, we were lucky that we would get some really, really small mice coming into traps. I think because there's not so many predators on the island, this might be why we get them. But that allowed us to look at, how the microbiome develops with age. And actually, the patterns are broadly similar. So if we look in lab mice, we see two clear patterns. As the mouse gets older, you get an increase in species richness, so their microbiome becomes more diverse. And these empty circles are before they wean from their mother, and then the filled circles are after they've been separated. So that's a very clear pattern. And the other thing you see is that they kind of develop a more adult-like microbiome. So if we look at how different their microbiome is to a juvenile microbiome, it becomes more different, and then it sort of stabilizes in this adult microbiome state. So that's the lab. In the wild, we don't have precise known ages, but we use body mass as a proxy, but we see broadly similar patterns, but a bit more muted, I guess. So we still see that richness increases as they get older to a plateau, and that they kind of converge on this adult-like microbiome, but it's a bit more gradual. So the patterns are similar, just a bit more gradual. And this may be because in the wild they're weaning slowly, whereas in the lab you just literally take the baby mice one day and off they go to be weaned and separated from their mother. And similarly, if we look at how the sort of key taxa change with age, there are some similarities. So in both the lab and the wild, we see, for example, these firmicutes coming down in the sort of early life, and the Bacteroides coming up until they're about equal proportions, and exactly the same in the wild. And these proteobacteria that you often see early in life, in both cases, kind of reduce to a very low level. So seems like there's some broad scale conserved patterns between lab and wild, and maybe this is because there's some sort of intrinsic program in how your microbiome develops. So when we then looked at what factors predicted variation within the population in microbiome composition, one of the patterns that jumped out extremely quickly was that there was very strong seasonal variation. So depending on when you catch a mouse, in which one of our field trips, so whether it's spring, summer, or autumn, you'll find a slightly different composition. Now they're sort of continuous with one another, but you can see in this ordination plot that you've got all the autumn microbiomes here, spring here, and summer here. And if we take this axis, which is sort of the primary axis of seasonal variation, and plot it over time, over the 4 years that we sampled mice in this population, you see this very clear oscillation. So you can think of the y-axis as an axis of microbiome compositional variation, and we see that every spring through to autumn, we see the same decline in this axis, and it comes back up again. So there's a predictable rhythm in the microbiome composition that occurs seasonally. And if we break this down and try and look within individuals, some mice will capture multiple times, so we looked within those mice to see whether this population level trend is happening within individuals, and it looks like it is. So in this figure, the lines connect samples from the same individual mouse, and you can see that essentially, at the times when you see these major changes, for example, between spring and summer at the population level, it's happening within individuals. So we think the seasonal changes within individual microbiome changes, largely. Okay, now on to the fun bit, I think. So this was kind of our big question. And when we saw this pattern of seasonal variation, we wondered, does this have any functional consequences? Is the microbiome doing anything different at different types of year? Maybe it's helping them adjust to a particular season. So we can start to think about, what this might be doing. And obviously, thinking about physiology, you've got to pick a trait. So how did we go about thinking about what trait to think about? I guess we thought about 3 different things. So we tried to focus on traits that we thought would matter for a mouse in the wild population on a windy island in Wales. We tried to think about traits that were thought to be influenced by the microbiome from lab studies. And we tried to focus on traits that would be sort of likely to influence this fitness trait. I think there was something else I was going to say there, but I can't remember what it was. Anyway, so what physiology might matter for a mouse on this island? Well, one thing we do know is that there's very high mortality in winter. So this is a plot of just the mouse numbers from an old study by a guy called Sam Berry and colleagues who studied mice on this same island in the 60s and 70s, which was actually really helpful for us in the end. So if you're doing science and you think, oh, I wonder if anyone will ever use my papers, read my papers, I think no one looked at this for, I don't know, 40 years, and then I picked it up and was like, yes. So what we see is that these big dips, these are overwinter population bottlenecks. About 70 to 90% of those mice die in the winter. And we can see the same sort of pattern in our more recent data. So we, as I said, we tag all the mice with this thing called a PIT tag. It's just a passive, that stands for passive integrated transponder. And whenever a mouse runs under one of our loggers that we place out in the field, we've had about 50 of those, it will record that it's present. And then we looked at the sort of survival curves of those tagged individuals after we left the island in October. So we just abandoned the island, come back in the spring, but you can see them dying throughout the winter quite clearly in both years. So we know winter's a big population bottleneck. And I guess we then wanted to think about what physiological traits might help the mice prepare for this. You know, winter is coming, everyone's going to die. Almost everyone. What could be important? And we landed on thermoregulation. So for a couple of reasons. Winter is obviously cold, and these old papers from the island by Berry and colleagues suggested that the overwinter mortality was associated with temperature in late winter. The stats on that were very, very shaky, but anyway. And they also suggested that mice show some physiological adjustment throughout the seasons. So as they move from summer to winter, this paper suggested that they reduce their resting metabolic rate, and that part of the way they do this is by dropping their body temperature. So this could be a physiological adaptation that would allow mice to survive those winters. And interestingly, recent microbiome research has also suggested links between the gut microbiome and body temperature, and also what you might call cold tolerance, how well a mammal deals with a cold challenge. I'll just give you one example of one of these papers because I think it's a really cool study. So this was a paper by Chevalier et al. On lab mice. And what they showed is that they basically had two groups of mice, one that was placed in a fridge-like environment for about a month, and one that was placed in a sort of room temperature environment. And then they took the microbiomes from those two groups and transplanted them into germ-free mice and looked at how it changed their physiology. And what you see is that if you give a mouse a microbiome that's been in a cold-exposed mouse, you see changes in their ability to maintain body temperature under cold. So you can see from these thermal images that these mice with a cold transplanted microbiome are sort of glowing, nice and warm, while the ones that had a room temperature microbiome are not as warm. So this provided some evidence that the microbiome in lab mice, at least, is regulating their response to a cold challenge. The cold microbiome also changed the size of fat deposits, so their subcutaneous fat around the leg and their perigonadal fat around the gonads shrank. And it also led to this really interesting effect where the white fat in the body started to turn beige. Now, what that means is that it's becoming more like brown fat, and brown fat is your heat-generating tissue. So that's how you undertake this process called non-shivering thermogenesis, so the ability to generate heat. So babies have a big patch of it on their back, and that's how they stay warm. Most of us lose most of our brown fat by adulthood, but not most of us, sorry, some of us lose most of our brown fat by adulthood. Some of us maintain little bits in regions here. So brown fat's a really important tissue for thermogenesis, and this microbiome induced browning of the white fat. In other words, supposedly helping mice deal with a cold challenge. So it's almost like the cold signal from the outside world is translated by the microbiome into physiological change. So this was really cool, and another reason why we thought, let's focus on thermoregulation. So we need to measure thermoregulation in these wild mice. How did we do that? So I had a PhD student and a postdoc, Carla and Holly at the time, who developed this simple assay where essentially we put mice in a warm environment as a sort of baseline for half an hour. And then we put them in a fridge for an hour. And we sort of take thermal images at 2 different time points to see how their body temperature changes in between. So we did these assays in sort of 3 of the later seasons that we went out to the island, and they, through a massive effort, did more than 200 of these assays. This is what it looks like in practice in the garage. So we've got our warm environment under this sleeping bag. It's kind of a grow tent that you, anyway, could grow plants in. And then you've got a fridge down here, which we put the mice in. And this is Carla taking a thermal image of the mice. So this just looks like this. We kind of let the mouse run around. And then you can analyze those videos to get their back temperature, their eye temperature. So most of the results I'll show you will be focused on the eye temperature because there's some evidence that the eye temperature from these images is a bit like a window to the brain. It's quite reflective of core body temperature. Alongside these assays, we also collected feces, and we tried to take as many samples in liquid nitrogen as we can. And the reason for this, so that's what they're doing with this dewar of liquid nitrogen, all this protective gear on, is that because that will preserve the microbes properly if we want to do microbiome transplant experiments, which was our ultimate goal. Okay. So what did this wild data on thermoregulation show us? So what it showed us was roughly what we expected, I suppose, although no one had ever looked at it before in wild mice. So the first thing it showed is that whenever you put mice, this is 22 degrees, this is 5 degrees, whenever you move them into the cold, they drop their body temperature. So you might think mice are a homeotherm, they should maintain 37.5 degrees, but actually they are facultative heterotherms. So they do drop their body temperature to save energy when you put them in the cold. So we saw that. But I'm just realizing my key's not come up, which is annoying. These different colors are the different seasons. So pink is summer, green is autumn, and yellow is spring. We also saw a difference in the sort of body temperature set point across seasons. And this was seen in the warm and the cold. So what you see is that these mice caught in spring have a body temperature that's about 1.5 degrees lower than mice caught in summer. And for anyone who wants to know which bits are significant, if you're a sort of, nerdy scientist, the stars mean that these are statistically significant differences. Okay, so we know that we have an interesting effect of season on body temperature, and we also know that the microbiome shifts seasonally. So what we wanted to do now through an experiment is ask, could this seasonal change in the microbiome be mediating these differences in body temperature? So we did this microbiome transplant experiment, and again, this was Carla, Holly, and they were joined by another postdoc in my lab, Alex. And in 2024, then they did this experiment where they took gut microbiome samples, these FMT samples from wild mice caught in summer, autumn, or spring, and they transplanted them into germ-free standard lab mouse genetic background mice. So these are just mice that have absolutely no gut microbes. So you give them a gut microbiome, wait a few weeks, and then you can measure different aspects of their biology. So in this case, we used this climate chamber that also can measure metabolic rate through something called indirect calorimetry to measure thermal physiology and metabolism of these mice that had a microbiome transplant. And I guess our hypothesis is that if the microbiome is driving these differences in body temperature, then we would expect the recipient mice to develop the traits of the donor mice. Okay, so what did we find? So this graph is just sort of a reminder of what I showed you before, but now with the key. So this is the differences in body temperature in those wild mice. And then over here, I'm going to show you what we found in the lab mice that we inoculated. So again, we've got the eye temperature as a measure of body temperature. This is the assay temperature down here. And now the colors are the inoculation treatment, so which microbiome they received. And strikingly, I think we all sort of, didn't quite believe this would happen. I thought it was made up, and we reanalyzed it and got somebody else to extract it, but we see a very clear recapitulation of this pattern. So when you give mice a spring microbiome, you just move the microbiome, they develop a body temperature that's significantly lower than if you give them a summer microbiome. What's that supposed to say? I have no idea. Doesn't matter. Anyway, it looks very similar, and actually even the magnitude of the differences is very similar. So it's about a 1.5 degree difference in the wild mice and a similar sort of difference in the inoculated mice. Perhaps even more convincing is that if we plot the body temperature of the donor and the body temperature of the recipient, we get this quite nice, strong correlation. So you can transfer body temperature with the microbiome, providing really good evidence that, it's the microbiome that is playing some causal role in shaping their body temperature across seasons. So that was cool. We then looked at some other traits that we perhaps couldn't measure in the field, but we could measure in the lab. So one thing we looked at was the activation of brown fat. And there are better ways to do this, which we'll do in due course, but the first thing we looked at was just to take our thermal images. We can measure the temperature of the brown fat by looking at the main deposit, which is between the shoulder blades. So we look at the temperature between the shoulder blades, and we say, what's the difference between that temperature and a temperature somewhere else in the body, like the hind leg? And that difference then is a measure of kind of brown fat activation. And what we find, sorry about these black squares again, is that we do see some seasonal differences in the level of brown fat activation. My slides have gone, I should have checked further down in these slides. What you can see though is that essentially spring and autumn microbiome have a higher level of brown fat activation than your summer microbiomes. So in other words, their brown fat is generating more heat for them in those colder months of spring and autumn. We also looked at overall energy expenditure. So this is essentially the chamber that we put them in can measure rates of oxygen consumption and CO2 production. And from that, you can calculate how many calories a mouse is burning per gram of their body weight per hour. And what we see here is that the mice that are given this spring microbiome or an autumn microbiome are expending less energy than mice given a summer microbiome. So this is probably driven by that drop in body temperature. You lower your body temperature and it saves energy. Finally, we had a look at the fat deposits. And the reason we were interested in this is partly because of that Chevalier paper that showed if you expose a mouse to cold, transfer the microbiome, their fat deposits differ in size. So mice have different fat deposits in their body. They've got these ones on the leg that I mentioned. They've got mesenteric fat, which is around the gut, and they've got these perigonadal fat deposits. So we looked at whether these differed at the end of the experiment in our microbiome transplant mice. And again, we found significant effects of these different seasonal microbiomes on these fat phenotypes. So the spring microbiome led to significantly smaller inguinal fat deposits, so these are the leg fat deposits, and smaller mesenteric fat deposits. And that actually mirrors what you see in lab mice in this Chevalier paper if you think that spring is the coldest season, which it is. It's sort of the overwinter season. But quite interestingly, I think, when we looked at this perigonadal fat, which is the fat around the gonads, we see a completely opposite pattern. And again, this is a very significant difference. So in fact, the mice given a spring microbiome develop larger fat masses around their gonads. Now that's actually not what the Chevalier paper showed. They showed that cold led to shrinking of these fat deposits. So this is a result we got, I think, last week. So I don't know what's going on. You're very lucky to see this. My suspicion is that, spring is when they're starting to breed. And these fat deposits are known to be important for reproductive output. If you remove them from mice, you get reduced reproductive output in males and females. So what could be happening is that the microbiome is somehow maintaining these perigonadal fat deposits because they're going to be important for breeding in the wild. So that's something we'll have to look more into. Okay, so I need to be quiet soon because I'm going on for too long. But just to summarize what we found then, if we compare what a spring microbiome of a mouse does to a summer microbiome, it induces a lower body temperature, lower energy expenditure, more brown fat activation, smaller white fat deposits, mostly, except for these ones around the gonads. What does this all mean? Our hypothesis, and we need to do more work on the sort of some of the mechanisms underlying this, but we think that the microbiome is setting these mice into kind of a low power mode. So these are the mice that have survived the winter, and maybe one of the reasons they have survived is that they've reduced their energy expenditure. I didn't show it to you, but we also have behavioral data showing that they're less jumpy and they move less. So this is our kind of working hypothesis. Future questions, we have many. Obviously, we're interested in what species are involved, how this is all happening. I guess one thing I'm interested in is what triggers the microbiome to change seasonally. Is it something that is sort of predictive, like day length, that mice are trained to, or is it more reactive? So I'm hungry, I have no food, I'm going to reduce my body temperature, or my microbiome is going to change to reduce my body temperature. And then, of course, the big million-dollar question is, does this actually have any impact on survival? The jury's still out on that one. I guess I wanted to just end by saying, this is just one species and perhaps not a particularly interesting one to many people, but there's growing evidence that the gut microbiome regulates physiology in lots of different species, and particularly seasonal physiology. So it seems to be involved in responding to predictable environmental changes. There's a couple of examples. So people have shown that the gut microbiome helps ground squirrels in maintaining their protein levels during hibernation. So the microbes recycle urea so that they can keep their protein levels up and stop muscle wasting. And a very recent study also showed in voles that, so voles shut down their reproduction if you put them in a short day length like winter. And recently it's been shown that the gut microbiome is involved there through fecal microbiome transplant as well. So really growing evidence that gut microbes are important in regulating physiology. And I think by understanding how microbes regulate sort of these predictable changes that animals have evolved to deal with, we can maybe also understand something about how animals might be able to respond to unpredictable environmental changes. So things like climate change. I think, it's highly likely that gut microbes may have a prominent role in regulating how animals are going to be able to deal with those increases in temperature as well. Okay, finally, I just have to thank everybody in my lab. So this was a big effort by a lot of people. They did a lot of hard field work with absolutely no guarantee we would find anything interesting at all. So big thanks to my group, Evelina, Carla, Alex, Holly, and Claire, who helped us with the experiments, and the wardens of Skocum, who've been incredibly tolerant of all our mousing around, even though they're mostly interested in seabirds. And thank you for listening.
All really great talks, thank you. A question for Sarah. I was wondering, do you see the seasonal changes across both genders?
Yes. Yeah, there's not really much sex difference, to be honest, in the microbiome change, or in the impact it has on physiology. A little bit in the fat, but otherwise not.
And I was just wondering if what mechanism you think, by which mechanism you think the microbiome is regulating fat deposits? Is it through mostly nutrition, or?
That's a good question, and I don't know. I don't know the answer to that. I'm not sure. I'm not a mechanistic biologist, if I'm honest, so I think I'd have to go dig into the obesity literature and see what those details are. But I'm not sure at this point.
This may sound ridiculous, but what about the devil's advocate position? What if the mouse is regulating the gut biome?
I'm sure it is. It definitely is. So it will have those microbes under quite tight host control. We know that mammals have lots of ways of controlling their microbes. So the oxygen levels in your gut are tightly controlled by you through countercurrent blood flow and things like that removes oxygen from the gut. So I don't think it's going to be as simple as the microbes are, in control and dictating mouse physiology. I think I see it more anyway as that the host has co-evolved with microbes for so long that it's using microbial signals as part of its homeostatic signaling networks. So that's my sort of view on it.
So do you think the microbiome influences the behavior of the mice? I mean, I'm just thinking of the possible changes in diet, for example, which are a matter of choice by the animals, which might affect the biome, but is stimulated by the mouse itself, surely.
Yeah, so that's an interesting suggestion that they might. So you're suggesting they might, as the seasons change, they might eat different things. The gut microbes change, and that it could be that that's what's happening. There had been a study recently that showed that if you transplant microbiomes from different species of rodent into mice, their diet preference changes. Where they go for more carb or more protein. So I think it's a possibility, for sure.
Right. Hello. I wanted to ask a question about, because you were saying quite a bit about how the populations drop in winter. Does predation from seabirds play into that, especially considering the one counter to that is that the mice that do survive are typically slower, you said? They're less active than the ones that don't survive winter?
No, we don't know anything about their behaviour. So the seabirds don't eat the mice. If anything, it would be the other way around, actually. The mice, they are known to predate seabirds in some islands. So on Gough Island, they're eating albatross chicks. It's really nasty. And they could eat the eggs and things. But there's not really that many predators on the island. There's the odd owl that flies over from a neighbouring island. But otherwise, it's mainly mice eating. They eat dead carcasses of seabirds a lot. So, I don't think it's predation. And that's one of the reasons why it's quite a nice system, is that it's unlikely to be predation that kills them in the winter. It's more likely to be food shortage and dealing with a nasty environment.
Yeah, a question for Theo. Have you been able to use the barcoding to, I guess, trace the progress of invasives to the UK, and I guess then potentially correlate that back to the impacts they've had in the places they've come from? So you showed you might be identifying potential pathogens. Have any of those potential pathogens developed into actual pathogens in France or Spain, or where those invasives may have come from?
Yeah, so we haven't looked at that data ourselves, but for example, one of the ones that we detect in the UK, Endoconidioptera, that's already causing quite big problems in Europe. One thing that we have seen in our data is already we see that some of these fungi that we thought were new are being picked up in other Ips species in the UK that are native. So we have a species called Ips sembrae, and it seems that some of the fungi that in the past have never been associated with Ips sembrae are where we find Ips sembrae and Ips typographus together. The UK native Ips sembrae has these new fungi. So it does seem that because these beetles often they will kind of pick up the fungi from their environment, there is some sort of vertical transmission that when the female travels to a new plant, they take fungi with them, but they also pick up new fungi from their environment. And as when they start to kind of cultivate these fungi, they're sort of free living. They're not like endosymbionts. Other beetles that are living in that area are able to pick them up. So whilst we don't know sort of the how it's going to progress in terms of kind of tree pathogenicity, we're already seeing that some UK native beetles are picking up these new plant pathogens, which is a bit worrying.
A two-part question, if you don't mind. One of them is about the temporal change in the richness that you showed. It's a very interesting pattern. I wanted to ask if you also have data from the lab in a more controlled environment, and whether or not you see the same seasonal fluctuations of the richness of the microbiome in a controlled setting in the lab.
I don't know if that data exists. So it's actually not richness, it's more like an axis of compositional variation. So it's not the number of species, it's, I guess the axis would be, it's hard to describe, but it's like when there's more of one taxa, less of another, it could be, yeah, it's just a compositional axis. But I don't know of any studies that have looked at seasonal variation in lab mouse colonies. So it's an interesting question whether they maintain that. But they are under, yeah, you would expect not. Okay. Otherwise, because you'd think, where are they getting their information that it's summer from if it's just on a 12-hour light-dark cycle and it's always 22 degrees. But that would be really fascinating if it stayed.
Thanks. And the second part of my question is about the FMT that you did, the microbial transplant. Just out of curiosity, how long do you wait after transplanting the microbiome for these effects to take place? Is it 2 days? Is it a month?
No, we left about a month, yeah. So they were about 4 or 5 weeks old when they got the microbiome, and then we waited 3.5 weeks, something like that. And it might be possible that we would need to wait longer to see some effects, for example, on their growth, body mass and things. We don't see too much effect on that, and it might be because we didn't wait long enough. But certainly, yeah, the thermal traits seem to transfer quite quickly and easily.
I have a couple of questions. First, Theo, I was curious if you know anything about, or if you're interested in anything that might kind of give more resilience to a forest, like of all the different kind of places that you sampled. Is there like anything like a monoculture plantation is more susceptible to an invasive, or like certain types of woodland are less susceptible, I suppose?
Yeah, I mean, we haven't kind of properly analysed our UK data set yet, but we would definitely expect that in a monoculture that they would be able to spread more quickly. I'm trying to think if there's anything else. Well, in general, kind of at higher temperatures, at least in kind of temperate regions, when trees are more stressed, they're more susceptible to bark beetle attacks, which is why kind of climate change seems to be a bit of a hot topic within the bark beetle literature at the moment. So the nice thing also about our UK data set is we have a sort of natural gradient. We have from down south all the way up into Scotland. So maybe that's something that we'll also be able to see. But because that data is quite new, we haven't looked at that yet. But yeah, I definitely think that forest composition, but also any factor that could lead to plant stress. So yeah, temperature or drought maybe as well.
Cool, thank you. That's really interesting. And then Sarah, I was wondering, this is kind of not strictly microbiome related, but I'm interested in whether you pick up microbes in kind of the gut sampling that you do that the mice might be eating. So for example, do you find, because I work on fungi, for example, do you think you pick up kind of signals of their diet? So, for example, if they're eating fungi, do you pick up fungi in their scat?
Yes, we do. Because when we did a little bit of ITS, fungal microbiome sequencing, and we definitely had to remove some mushroom sequences from there. So there is some in there.
Very interesting.
Yeah.
Cool.
You expect that though, I guess, right?
Yeah, I've been like talking to some people about, I work on mycorrhizal fungi, about whether anyone's like looked in the UK at kind of detecting those fungi in rodents, wild rodents, and kind of struggling to find anyone that's actually working on it. But it's been shown in like America and Australia.
Yeah, I'll dig out my data, you can have it if you want.
Yes, please.